I am Sanchari Mukhopadhyay from University of Calcutta presenting about pollination biology of different buzz-pollinating bees in an agricultural landscape in eastern India. We are all aware of hive-building social bees, but I will be talking about the solitary bees, particularly capable of vibrating their thoracic muscles to release pollen grains from the structurally modified poricidal anthers of Solanaceae flowers. These bees are called as the buzz pollinators, and while studying their behavior in Balasore, Odisha, I recorded 13 buzz pollinator and 3 non-buzz pollinator species visiting brinjal flowers, all for the single limiting resource pollen, which makes competition inevitable. 44% of the interspecific interactions were observed, of which 81% were provocative, but the attacker bees showed aggressive behavior towards the defender bees. By calculating David-Score dominance hierarchy, Amegilla, Nomia, and Xylocopa were found to be the most dominant in the community. However, such harsh competition have not led to the exclusion of other bee species from foraging in the brinjal flowers, which indicates that there might be a foraging preference for the bees to avoid competition. The brinjal flowers showed high variation in their morphological structures, and a bee flower network with high modularity showed that bees formed different groups with similar types of flowers, showing preferences for floral traits guided by flower diameter, height of stigma, anther length, and anther width. Thus, they do compete yet coexist by partitioning the resource. But why are non-buzz pollinators like honeybees who cannot buzz are present in brinjal fields? Experimental study with the most frequent buzz and non-buzz visitor species confirmed that honeybees visit newly bloomed virgin flowers for about 10 seconds, which increases 2 to 4 times, succeeding a single visit by a buzz pollinator species, and their increasing number of buzzes, which probably dislodges pollen grains from the anthers, making them available to the honeybees through the apical pores. The bigger bodied Xylocopa was found to be a better facilitator compared to Nomia. However, the pesticide was found to be acting as a stressor by altering the buzzing efforts of the buzz pollinator species based on their body sizes. In agricultural fields with high amounts of chemical pesticides, large-sized bees showed a notable increase in their sonication frequency. Medium-sized bees showed a notable increase in buzz number, buzz duration, and sonication frequency, whereas small-sized bees showed increased buzz duration but significantly decreased visitation duration, buzz number, and sonication frequency. This whole study is significant because it first documented evidences of a dominance hierarchy among bee species competing for a single limiting floral resource in a brinjal field, but it is a pioneering attempt to measure sonication frequencies in tropical buzz pollinating bees in India and demonstrates that buzzing behavior of pollinators facilitates pollen access for non-buzzing honeybees in poricidal anthers. It also reveals that pesticide exposure impairs sonication performance, placing medium and small-sized buzz pollinators at a competitive and energetic disadvantage.